Welcome everyone to my talk. Thanks for being here today. So my name is Hannah. I am from Anna Dragoš's lab in Slovenia, and I'm here to talk to you about some new microscope pipelines that we're coming up with. So the best way to convince someone of something is to show it happening. And a fundamental piece of knowledge that everyone here would have is that when phages are floating around in the environment, they eventually land on a bacteria that it's its host. It will then go through a cycle of attachments, infection, and host cell lysis, releasing progeny phage. And from that fundamental piece of knowledge we have, that was generated from multiple different types of experiments, from different microbiology, molecular biology, technical assays, as well as microscopy techniques. However, these ranges of experiments have different spatial and temporal coverage. So the goal that I've been working on is to develop a new pipeline for looking at bacillus cell and phage interaction at the single cell level over a long period of time, so a high spatial and high temporal coverage. So we have access to a microscope in our lab called the ELIRA 7 microscope, which is a very fancy, expensive piece of machine. It's super resolution. That means it has a SIM2 capability down to like the 60 nanometer resolution. It also has tracking, dynamic tracking processes in both 2D and 3D over a long period of time. We can look at multiple fluorescence colors and also look at, also integrated with the many other features. So the pipeline that I'll be showing you is a new pipeline for looking at the fluorescence of bacteria, phage, and bacteria in this pipeline, which is made up of four different sections. We have sample preparation, slide preparation, microscopy setup, and imaging processing. So the first part of it, which is the sample preparation, with our first experiment, we used a Bacillus subtilis phage called NF, which is a lytic phage and can produce a quite high titer. We have stained this phage using a stain called cyber gold, which produces a green fluorescence when it's attached to DNA. So this is a DNA binding stain that only fluoresces when it's bound to the DNA. The bacteria that we're using is B. subtilis, which is a very well-studied bacteria. Part of the reason why we're so interested is because we know certain phages that can manipulate its host, the B. subtilis. It has high ecological relevance and a very high industrial relevance as well. So for this experiment, we used a lab strain called P9B1, which has been genetically engineered to constitutively express a fluorescent protein called MCATE, which produces a red fluorescence. So we're looking at the red and green today, almost Christmas. So slide preparation is very straightforward. So the thing with this is because we are looking at bacteria, we need to create a good environment for these bacteria to grow. So using a thermoscientific gene frame, which is a blue frame that adheres to the glass, we can generate agarose pads that we then cover with a polymer cover slip. The slip in this looks quite large, however, it can be cut up into sections. So once we put all the components together, we have 1.5% agarose pad, and for this, we're using M9 media. So one of the reasons why we chose M9 media over, let's say, LB agarose, not LB, yeah, LB agarose, is because M9 is a transparent media, so there's not much background fluorescence. So the samples are added to both sides of the pads, and they mix in the middle. So another addition about the cover slip is one of the reasons we use this cover slip is because those who work with bacillus know that it will not survive at low oxygen levels. So this is a oxygen permeable cover slip that we use. Next is the microscope setup, which we use Zen Black software. It's all very straightforward. We set the temperature for the incubation chamber at 32 degrees, so the cells are growing quite happily at that temperature. We use the red and green laser at power level and exposure times that we have tested to see which exposure gives the best signal, but doesn't damage the cells too much. For the time lapse, we are doing 300 cycles. So every 3 minutes, there is a image taken of the red and green fluorescence. It works out to be about 15 to 16 hours coverage. but we can set it for longer if we want. So the final process is process of this pipeline is imaging processing, which is quite an important step. So because I mentioned before how we're using the M9 media background, how it still produces, the M9 media, although it's transparent, will still produce a small amount of background fluorescence, which will be in addition to the signals that we see. So we had to go through a round of subtraction. So from the red channel, we subtract any red background that could be there, and we do this with the green channel. However, unfortunately, we noticed immediately that an issue with the system is because the MK signal from the bacteria is so strong, there's actually crosstalk in it with MK crosstalking into the cyber gold channel, the green channel. So in addition to removing the background from the green channel, we've also had to develop a system to remove the MK crosstalk. That's where we came up with a control region scaling approach method, which was used to scale the red background in such a way that it could then be subtracted from the green channel, removing the crosstalk. An additional issue that we noticed immediately is while the phage samples were thoroughly washed and filtered to remove as much excess stain as we could, there is still a small amount of unbound stain remaining in the sample, although not much at all. So unfortunately, with this stain present, when the host cell lyses and releases its genomic DNA, the stain can bind to it. So these are just some control images I want to show you first before we get to the time-lapse results. On the left-hand side, you see, actually, can we turn off? Yeah. On the left-hand side, you can see Bacillus subtilis producing a nice red signal. On the right is the NF cyber gold stained phage. So as you can see here, unfortunately, because the stain is bound to the phage DNA, which is encapsulated in the head, the signal is not very strong, but you can see dots that are all about the same size spread around the pad. You can either see some larger dots, which is most likely the phage that has aggregated with one another, that can sometimes occur during the filtering process. However, this I feel is not a very accurate representation of what the phage look like on the pad. So this is just phage that a droplet of phage in SM buffer has been added to the pad and left to dry. What I found the phage look more like is, now it's very hard to see with this much light in the room, but if you look closely, you can see green flashes coming in and out of focus, which is a better representation of what the phages were doing on these pads. Rather than just nicely sitting on the pad, although some were, the phages, probably because of the size of them, were quite mobile and coming in and out of focus. So unfortunately, this does make it tracking them a little bit harder, but we're not going to let that stop us. So finally, time lapses. So I'm just going to play it for you now and not say anything so you can properly watch it without trying to listen to me. Yeah. So this was with bacillus and phage added at a 1-to-1 ratio. So there's a lot happening. So to better analyze what's specifically happening in this time-lapse position, because we can look at multiple positions, I'm going to direct you to this specific square. So I'll just play it again once. So we're going to take a quick step-by-step look at what's happening in this specific section of the time-lapse. So within the first about 54 minutes or so, is it playing? No, sorry. Yeah. Okay, so unfortunately the first time, this time-lapse, wait, no, yes. So within the first 54 minutes of the time-lapse, the cells are growing and dividing as normal. And I might remind everyone, the samples were added together. So for the most part, within the first 54 minutes, the cells are growing normally and happily, living their best life. However, at about the 57-minute mark, we see a yellow dot presenting itself in one of the cells. So this is the red MCATE and the green are now overlaid. So there is phage in that cell. After about 12 minutes, the green signal has now become more apparent, and the red signal has almost been completely depleted from the cell. About 39 minutes post-infection, it is estimated that the cell has died and lysed, and now the cells surrounding it are moving in as cells around it divide. So the cells above and below are pushing inwards. So you'll notice that there's a lot of green, there's a thick green dot, and sometimes around it, there's a lighter green halo. So at about 6 minutes after the death of the initial cell, the cell directly to the left has disappeared. It might be a little hard to notice. Yeah. So this cell, adjacent left, probably the closest, has more or less completely stopped having any MCATE signal. About 42 minutes after the cell has stopped showing the MCATE signal, a small green cloud has formed where it is. And a further 45 minutes after the death of the initial cell, the cell directly above the initial cell has also stopped producing any MCATE signal. And this ultimately also leads to a lot of cell deaths in the surrounding area. So just quickly summarizing what we watched. Within the first about 39 minutes of the time lapse, 39 minutes, we see a yellow signal pop up, which is an overlay of the red and the green. Then over time, the green signal goes down, or the green signal appears more vibrant, and the red signal is slowly going down until there's just a green signal left. Sometimes there's also a green halo surrounding the cell. About 40, after the death of the initial cell, with the cells pushing into this new free space, we then start to see the surrounding cells begin to die as well, although they're not producing the same sort of vibrant green cell that's initially seen in the first set of cell lysis. So about 42 minutes to 45 minutes, a lot of the cells surrounding that initial cell have begun to die. So in conclusion, and to take you back to the start, a pipeline that enables real-time single-cell visualization of Bacillus subtilis interactions at the nanometer scale is a partial success, to this point at least. So we've achieved a number of things in the development of this pipeline, including partially being able to track phage, being able to observe phage absorption, host cell lysis, and we've also resolved the issue of the strong crosstalk signal, and infection. We've been able to track infection progression over real time. However, further development for this pipeline is still required, specifically removing the leftover DNA stain in the sample, as well as attachment. Observing the attachment of phage is still a bit limited since it requires the phage to kind of sit on the cell at the right time and at the right position. And finally, we can't observe the progeny cell that are not stained in this sense. So for future direction for this assay system, we're looking at potentially developing genetically modified phage that have fluorescent tags labeled. This would solve the issue of the leftover stain in the sample, as well as give us the ability to study the progeny phage and track them, or track them better. As well as we're going to be looking at performing these assays using the super resolution imaging, specifically SIM2. Now, the reason we haven't done that yet is because SIM2 is much more intensive imaging on the cell, so it's a bit more damaging to the cell. We wanted to make sure the pipeline was working, and it also produces an incredible amount of data. So we started off with just the wide laser field. And finally, with the images that we do have of the time lapse, we want to use other tracking software to track the fluorescence of both the bacteria and phage, and see if we can actually get a better understanding of how the bacteria will react when there's phage present, diverse when there's no phage present. Finally, a massive acknowledgement to everyone in my lab who's helped me with this pipeline. And thank you so much for listening to my talk.